well, we can get started. Welcome to this session on bioinformatics workflows and core facilities. We are grateful for sponsorship from Scale Biosciences, and Santiago Salas will be giving us a presentation to start the day off. Santiago has been working in genomics for about 15 years and is really keen and eager to bring fancy tools to the research community. So welcome to the stage.
All right. Well, hello everyone. Thanks for joining the session. I know you're not here for me, so I'll be quick. Thanks, Charlie, for the intro and for the panel. So yeah, my name is Santiago Salas. I am a sales specialist with a company called Scale Biosciences. I am going to talk 10 minutes. I promise it's going to be short and quick on a tangential topic for what you guys are here. It is not a heavily bioinformatics talk for me. But I hope you guys will enjoy it. Although there's been amazing progress in single-cell omics throughout the last few years, as we all know, thousands and thousands of publications and data sets, we believe that we're just scratching the surface of understanding biology, and specifically single-cell biology. And so our mission is to lower the barriers and the perceived barriers of single-cell biology, starting with experimental design and cost constraints, whether it's on the kit configuration and technology constraints. We really want to enable people and researchers to think outside the box and let their experimental and biological needs dictate how the experiment is or takes place, rather than technology and legacy developers dictate that for them. And so, as we focus more on lowering the sequencing costs and scaling up the sequencing capabilities with partners like Ultima, we believe that single-cell sequencing will continue to be a driving force in science. So most of you don't know about us. We're a fairly young company. We've been around for about 2 years. In those 2 years, we've already quickly launched a number of products along this continuum, starting with transcriptomics, epigenomics, which is we have a really exciting single-cell whole genome methylation product that's very unique to us. Proteomics, where we have a total-seq phenocyte product that we collaborate with BioLegend, and 2 specific products on the transcriptomic side that I will highlight today for you. So I will start with ScalePlex, which is a way to pre-tag samples, pre-index samples before going into a single-cell RNA library workflow. This is similar to what you guys already know with hashing, but instead of being a lipid or a antibody hashing workflow, it's a simple oligo workflow. And it has many advantages. I can't go into all of them today, but it's a very unique way in maximizing sample input before going into these workflows. And because it's oligo-based and not antibody or lipid-based, it requires very minimum washing and steps, and it's very amenable to automation as well. The second product that I'm really excited to talk about is something we released at AGBT just a few weeks ago. It's our new quantum scale RNA platform. And so digging into the workflow a little bit, we start with cell fixation still with either ScalePlex, which I just mentioned, and you can do 96 samples at a time or individual samples, and then go straight into a reverse transcription plate, which is nothing new in generating cDNA, obviously, right? And then going into what we call a quantum barcoding plate, which is where the magic happens and where the big innovation in our product lies. And so it's almost like a hybrid, and I'll double click in the next slide on this specific step. And then you finalize by indexing, by adding PCR indices for either Illumina or Ultima libraries. And this would be in a native form, right? There's no library conversion to go into Ultima. I guess this is a slide where, I'll take an extra second since it's the topic of this session. Our data analysis pipeline does simple demultiplexing, genome alignment, transcriptome alignment, and others to provide a QC report. It generates barcode, knee plots, gene expression count matrices per sample, and gives you output that you would then put into a Seurat or ScanPy or any other pipeline to generate UMaps and other data. And so all of this is packaged together in Nextflow. And so, yeah, I said Nextflow, and I earned my spot here today. So, okay, so double-clicking on that quantum barcoding plate. So it's a modified 96-well plate. It has obviously 96 wells, but within each one of those wells, there's thousands and thousands of microwells where the cells are deposited after that RT step. And then we add a barcoded bead pool that increases the barcode space to the tune of 600 million barcodes. And that's how we maintain that very low recovery rate, very high recovery rate and very low multiplet rate that's actually standard in the industry. That is regardless of whether you're doing an 80,000 cell experiment or a 4 million cell experiment. That multiplet rate stays the same. And so on the product side, we have a bit of a continuum of products. So it's all the same chemistry, and it's all the same kit or plate that you're buying, but you're using different wells within that plate to extend the throughput of your library preparation needs. So you can go from 85,000 cells output or nuclei of libraries generated up to 4 million cells. And you can see here that we have, we've added the Scaleplex samples to show how really 4 million cells divided by a large number of samples can be very enabling for high throughput screens, CRISPR or other drug screens. And I'll show a couple more examples of how that comes along. But the pricing, we're definitely lowering the pricing for single cell applications. For those of you who are in the wet lab, and I don't know if there's many of you here today, the time of processing 84,000 cells or 4 million cells is basically maintained. There's a couple hours differences because there's a little bit more pipetting, but it's not an exponential or a linear increase in time. So it is very friendly with wet lab people, who we all depend on here, right? I mentioned a few examples of this, of how the kit can be utilized. So one can think of a proof of concept study with 85,000 cells, 4 samples, 20,000 cells, and these are the costs either per cell or or for the kit. You can go what we call instrument free, and this is directly matching what the current technologies can offer, right? 8 samples to about 10,000 cells per sample, and a lot of people like to push that and overload the cartridge. And then you can go to a 2 million cell output for a larger patient cohorts, 384 samples processed at once at the same time, 5,000 cells per sample. And then you can do massive screens like I mentioned before. I like to highlight this slide and acknowledge a vast community of researchers. So we launched a 100 million cell initiative together with some partners down here. And just wanted to, so we announced the winners back at AGBT, and it's just seeing the sheer number of applications tells you, or told us, how much this was needed in the field, right? So I invite you guys to check out the applicants that won, quote, the projects, and they've been supported by us, CZI, Ultima, and NVIDIA. We also have a bunch of partners in the workflow continuum. You probably talked to a lot of them today or the next few days. And lastly, because this is so short and I can't really show you a ton of data, I invite you to visit us either at our booth, 525. We're there through Wednesday. But most importantly, we have a mimosa bar tomorrow morning with breakfast. And we'll have a conversation around single-cell RNA and a little talk from our partners at Ultima Genomics as well to show what that data looks like in their hands. And with that, I will wish you good luck in Vegas, and thank you for your attention. Yeah, no worries.
Have you noticed any different cell types that perform differently in the quantum barcoding step?
Yeah, so I mentioned the 100 million cell initiative. That is, quantum specifically is new to the market, right? So it's only been around for a month. So we're still exploring. I can tell you that we can't really talk too much about it, but all those initiatives had a lot of different sample types. Mostly mammalian cells for now, for validation and all that, but we expect it to be compatible, just like our previous kit, with plants and other organisms in general. Yep. Thank you. Thank you.
Okay, little IT switch over here. Yeah, so thank you all very much for coming to this session. This session is something that was generated in the Genomics Bioinformatics Research Group. I'm involved in that group. It's the topic of our research group activity for this year. And so that's where this is coming from. I lead a Bioinformatics Core at the Koch Institute at MIT. And I'm very excited about these workflow managers because of the impact that they've had on me and my core facility operations. I think we're doing a much better job for people, providing them with access to the information that comes out of these workflows. The scalability and reproducibility and computational efficiency that they bring has really made a big difference for our operations. The reason why these workflows have been so easy to adopt is largely driven by the NF Core organization and all the community-driven workflows that exist there. It's a great way to get familiar and start executing best practice type bioinformatics workflows very quickly. And one of the things that's most exciting to me is that my novice computational users in our community are able to pick these things up and work with them pretty quickly. So NF Core is written in Nextflow, and as part of our research group study, we did a survey of our colleagues, and one of the messages from that survey was that this Sakara, Nextflow, NF Core ecosystem is pretty important in this space. So we're really focusing in on that. So we have 3 speakers today. Oops. Looks like it took over. Okay, great. Oh, maybe it's coming back. Nope. Okay, got it out of the way. Sorry about that. 3 speakers today. First speaker is going to be Ken Brewer. He's Senior developer advocate at Sakara. He has a PhD in RNA biochemistry and computational genomics and extensive experience migrating workflows from legacy HPC environments to more modern computational solutions. The next speaker after that will be Ramiro Barrantes-Reynolds, bioinformaticist and statistician at the UVM Bioinformatics Core, also has extensive experience in industry. He's affiliated with the Department of Genetics and particularly interested in infectious disease. And finally, I meant to say Ramiro is a member of GBERG as well. And finally, Lorena Pantano, Director of Bioinformatics Platform at the Harvard Chan Bioinformatics School. I know Lorena was a very early adopter of bioinformatics pipelines and workflows and has over 13 years of experience providing support in life science research. And Lorena is the co-chair of GBERG. So a little bit of an advertisement. GBERG's part of ABRF research group and welcoming new members. Anyone interested can just get in touch with me. Okay, so we can get started. Let's see. Do I go all the way to the bottom?
Yeah, that's fine. Okay. Thank you so much, Charlie. My name's Ken Brewer. I'm a developer advocate at Sakara, which is the company founded by the creators of Nextflow, continues developing Nextflow, and as part of my role, I do extensive outreach both to the academic community and also to the open source community. So I'm really excited to be here. The question that I kind of wanted to begin by posing is bioinformatics more of a science or an engineering discipline? And to answer that question, I want to take a step back and look at another science to engineering transition. And we're going to go back to 1860, when peak science at that time was figuring out how the internal combustion engine could possibly work. And here we have Etienne Lenoir, who was really excited about electroplating and figured out that you could use an ignition coil to trigger coal dust in a cylinder to create the forces that you need to drive a cylinder forward. About 30 years later, the internal combustion engine had moved into its artisan phase. And here are some of the greatest craftsmen of the artisan era of the automobile, René Panhard and Émile Levasseur, who were French artisans who developed a lot of the standards that are still used in modern automobiles today. Things like the steering wheel, a transmission with a clutch, a front engine, rear-wheel drive, were all developed in the process of doing one-of-a-kind automobile builds, mostly for wealthy European aristocrats. Now let's jump up another 20 years, and this is kind of peak engineering for the automobile, at least in this era. And this is when automobile manufacturing had moved to the stage where the focus was building them efficiently at massive scale. And Henry Ford, of course, is the person that we think of as the person who brought a lot of these innovations to the market. Well, one detail that I think is incredibly cool is that from the time when metal ore was being pulled out of the mountains to when it was making its way into finished automobiles, some of the Ford factories could do that in 80 hours. Talk about turnaround time there for a really complicated process. Now, I think kind of the answer to that question, thinking about all of those, is that we're a field in transition, and at least in its current state, I think bioinformatics is a science, an artisan craft, and an engineering discipline. And that's part of what makes it so challenging, and especially in core facilities, you're kind of working at the interface and the boundaries of these different types of challenges that you might face. And so what are some of the lessons that we might apply from learning about how automobiles were industrialized that I think could work for bioinformatics? There's three things that I want to focus on that I think have some applicability. The first is the importance of building with the right tools. So using the right tool for the job instead of solving every problem with another bash script. The second is the importance of reusing instead of reinventing. And so don't reinvent the wheel when there's a good solution, and try and offload your undifferentiated heavy lifting where possible. And the last one is the importance of scaling with automation and automating routine tasks where you can, and just reduce that turnaround time to kind of chase that 80 hours, except hopefully a little bit shorter, that Ford was able to achieve with his metal ore to automobiles. And I also like these kind of lessons because they fit really well with the mission of Sakara, the company. We view our mission as advancing science for everyone through software. And we like to organize that into three pillars for different pieces of the bioinformatics tooling and ecosystem that we work on. In the build section, we're the company behind Nextflow, MultiQC, and Wave. We have products for making it easier to discover things and not reinvent stuff that already exists, things like Sakara containers and pipelines. And then we also have tooling that makes it much easier to scale your analyses and automate things. So let's go to the first idea that I had around building with the right tools. Choosing the right tools is particularly challenging, in part because you're always moving between these two kind of extremes, where sometimes you need to kind of work a little bit more as an artisan, where you're a single researcher doing exploratory work, and you're rapidly changing your approach, tools, and parameters. And then on the other extreme, something more similar to what a large team might want, where you're engineering things and building production pipelines and data pipelines at enterprise scale. And ideally, you want a tool that can kind of journey with you and move from when you're in the artisan phase all the way to the engineering phase. And you want something that can be both readable and portable and scalable and reproducible, but isn't a pain to work with while you're still like iterating quickly. And I believe, and I think some of the other people here on this stage as well believe, that Nextflow is that solution. And Nextflow, it does it all, and it's open source and completely free for anyone to use, and it runs anywhere. And kind of the way that it achieves it is by having a very powerful command line interface that you can use to run programs that are written in the Nextflow language. So Nextflow is both a piece of software and a programming language. And Nextflow allows you to connect your code repositories, where you're storing that Nextflow code, together with all sorts of different compute environments where you might want to run that pipeline, or where your collaborators might want to run that pipeline. Anything from different cloud executors to Slurm and PBS, and a wide range of different ways of actually executing the tasks that need to be done. And it handles the parallelization and scaling for you, and it has a rich developer like toolkit around it that makes it easier to build Nextflow pipelines. Now, of course, writing Nextflow and needing to learn a new programming language to develop in Nextflow is a tall order. And so something that Sakara introduced recently is Sakara AI. And we wanted to tackle the challenging problem of how do we generate code that actually works in bioinformatics. And if you've tried to get ChatGPT or GitHub Copilot to write Nextflow code, you'll be familiar with the fact that it really struggles, in large part because of a syntax change that happened in around 2020 for Nextflow. And Sakara AI does this really well. And here's an example. Can you write me a process that uses GATK haplotype caller to call variants on the whole genome? And Sakara AI will output a process that follows kind of NF Core best practices. And after it's done, it will give you a little button that says, do you want me to test this code and make sure that it works? And if you click yes, it will go through the process of checking the code, finding a sample data set and testing it to see if it runs successfully. And if it encounters an error message, it will read that error message and then rewrite the code until you get something that works. So it gives you a much more reliable way of getting to a correct answer quickly. And so this is available today at sakara.io/askai. And we have a generous free allowance for people to experiment. And it's really good for converting any bash or other language scripts into Nextflow. It helps really well with like AI error debugging and code testing. And it's really rooted in NF Core best practices because it's been it was developed and tested extensively both by Sakara and a lot of our collaborators within the Nextflow and NF Core communities. So the second lesson from manufacturing that I wanted to talk about is the importance of reusing and not instead of reinventing. And this takes us straight to the NF Core organization. And NF Core is an incredible open source, completely independent organization that was put together by a collection of core facilities, originally in Europe, who wanted to use this incredible, very portable technology, Nextflow, to write bioinformatics pipelines that they could then share amongst themselves. So instead of having every core facility have their own way of doing RNA-seq analysis, can we put our heads together and come up with what's the gold standard that it should be and write it in Nextflow so everybody could use it? And it does that quite effectively. And they also have great graphic design with these metro maps that show how the pipelines are generated. This is NF Core RNA-seq. And it also illustrates quite well the complexity that Nextflow is designed to help solve, where you have some like 20 to 30 different command line tools that need to be stitched together, and where you need to be able to select which aligner you want to use, what type of different kind of like pseudo aligners you want to work with. And Nextflow is able to track all of the inputs and outputs from all of these steps and seamlessly pass things through to give you a reproducible answer to how you're doing the analysis, despite a very complex amount of software that's involved in producing that answer. Now, NF Core has over 100 pipelines that are available out of the box. And these range, they're primarily bioinformatics focused, but they also cover things like geospatial imaging and gravitational wave detection. Beyond that, there's also a large library of sub-workflows and 1,400 modules wrapping some of the most popular bioinformatics tools. So when you start developing a Nextflow pipeline that uses some sort of standard tool like FastQC, you don't need to go and create all of the boilerplate Nextflow language around using that. You can just import or install that module using NF Core tooling, and you'll be able to start using that command line tool right away. So that's about NF Core, and I think it's probably the primary driver around how Nextflow is able to enable organizations and companies and cores to reuse instead of reinventing things. And it would be great to have even more people contributing towards these pipelines to make them even better. Now, the last thing that I want to talk about is the importance of scaling with automation where possible. The solution that Sakara offers in that front is called Sakara Platform, and it's intended to be a single platform for the entire scientific data lifecycle. And it's connected deeply with Nextflow. And essentially, once you have a Nextflow pipeline, you can simply connect your code repositories to Sakara Platform, and then along with your Nextflow pipeline, which is just a CLI tool, all of a sudden you have a beautiful user interface for your wet lab scientists. You have API endpoints that your automation engineers can use. You've got CLIs and automation events that you can use to trigger a pipeline to run as soon as data comes off the sequencer. And I used both Nextflow and Cycera platform extensively for 3 years before joining Cycera about 6 months ago. And so I can speak from my own experience of how well it works in both very small organizations and much larger ones at solving a lot of these headaches. And kind of some of the things that you get with that, like I mentioned before, is you can get very user-friendly UIs that allow sort of non-computational people to launch these complicated Nextflow pipelines independently. And once they've launched something with like a very easy click-through form, you've got detailed logging and collection of the information in a centralized place. This includes metrics like cost estimates, how much of the CPUs are being utilized, and makes it easy to do optimizations around making sure you're not over-requesting too much compute resources for your Nextflow pipelines. It also makes debugging far easier, especially in distributed compute environments where the logs for a given task might be failing on node number 246 of who knows how many. So now, the one thing that I especially want to call out to this audience, and a big part of why there is only a member of the community team here and not anyone from our sales organization at ABRF, is we make Cycera platform completely free for any academic users, and including core facilities. So at degree-granting academic institutions who are doing non-commercial research, we have an academic program where you can submit an application and we'll give you a free Cycera Cloud Pro account, although that comes with the caveat that it doesn't come with professional support. So those were the core things that I wanted to talk about. So just coming back once again to these lessons from the automobile era that we can use for scaling our bioinformatics. I think it's important to be building with the right tools, reusing instead of reinventing, and using tools that allow you to scale and automate where possible, so you're not spending so much time on routine tasks. And thank you, and I can take some questions now.
Yeah, I meant to say that we're going to try to take questions after each talk, so you can just come up to the microphone if you feel like it. I was wondering that you mentioned undifferentiated heavy lifting and how to offload that. It sounds really appealing to me. I wondered if you could talk more about what undifferentiated heavy lifting is.
Yeah, so that's a catchphrase that I stole from AWS and Amazon. That's part of their philosophy of how they develop tools in terms of identifying, hey, all sorts of businesses have these types of headaches that they need to solve. What products or services can we use to offload those headaches? I think one of the main important things in that front is to be aware of the technology solutions that exist around you, especially in the open source front. A lot of times when you start building, it's easy to kind of get your head down and not look up above what's happening in your institution. And there's a lot of incredible open source tools that get developed and are released, and that sometimes have pretty mature adoption that can solve some of those headaches. So I'll mention, for example, Quilt as an example of something that can be used to kind of like give you like data repository like metadata that you can store easily with your data that you generate. And that's something that there's actually a Nextflow plugin for that makes it easier to run Nextflow pipelines and generate that metadata.
Great, thank you. Any other questions? Okay, thanks a lot, Ken.
Okay, and one last call out, the Nextflow Summit is happening in Boston this year in May, and we have a special 20% off discount for people who would like to attend. That's Core-20. So, thank you.
I think so.
It should be still open.
Oops. Okay, yeah, so my name is Ramiro. By the way, I'm from Costa Rica originally, and actually both of my parents are biologists, and I would like to think that I learned to collaborate from them biology, maybe not when I was a teenager, but now it's, I like to learn that I learned a little bit over time. And this is what I'm going to talk about collaborations in Nextflow, actually continuing on the thread that Ken started with Nextflow. When I saw this and I said, like, this is something that could be really useful in collaborations. I work in a core facility, but my role has really been in projects, interacting with the various core facility people and dealing with their projects. So I'm just going to talk about different ways to do it, okay? So we can go all the way from where I do everything in the project, you know, and I give them the end result, to where they do everything. So this is what I'm going to talk about. I do everything versus they do everything. And if you are in a small core facility like I am, it's useful actually when the other person does everything or most of the work. Right, so this is what I'm going to be talking about, and that's where Nextflow and NF Core became very important. So the one extreme is I do everything, okay? So this is Mr. Ben Lee. He's studying children with diarrhea, and in African countries, that's a really big issue, actually, okay? So he wanted something to be able to type various viruses there. So this is a pipeline. I'm not going to go through it, but there's various steps in the pipeline that take you all the way from all the viruses that he did in Nanopore. He sequenced in Nanopore, and it was terabytes and terabytes of data over the span of many, many months. And at the end, you have the types of the viruses that are there. Okay, so that's one extreme. And the Nextflow pipeline would be in the big brush terms will be like this, and there's obviously more to that, but what I want to say is that it's possible. I'm not saying it's easy, but it can be done. And mind you, by the way, in this project in particular, something happened, which is that we discovered that there was a new and shiny database that could be used for this, right? So we had to run everything again, all the terabytes of data, everything again. So with Nextflow, it was actually not too bad to go and do it. Right, where, you know, just everything's kind of set up so in containers and you know, pipelines and all that, and you can just run it all. Again, so yeah, so that's one extreme. Me doing everything, right? So now the other way it can happen is actually with you have the NF Core pipelines, like Ken was talking about. You have all these NF Core pipelines. Before I continue, actually, just a show of hands, how many people here use NF Core and Nextflow? Yeah. Okay, so several of you, right? Okay, so, and I'm just curious, you know, how many people use it, like the way that I'm talking about, you're interacting with somebody, and you use them to create a pipeline, and you give a deliverable. Yes, several people, right? So, yes, okay, so that's, the other thing is the NF Core has all these existing pipelines that you can use, and you run them as easy as this, like this, and there's even easier ways of doing it. And it's actually something that I can go and teach a collaborator to do it. If they have some modicum of skill, they might even be able to do this, right? The problem is that in my experience, it's never so simple. It's never as simple as getting an existing pipeline and running it. There's usually a lot more involved, right? It's nothing is kind of straightforward usually in bioinformatics. That's been my experience. So the other thing is you can do is we build up an existing pipeline and add other steps that we want to use, okay? So this is another example. Emily Bruce, she's another researcher at UVM. And she had another viral project, in this case, influenza, and another typing project. And this is a pipeline that was actually developed by somebody at the CDC. He wanted to do something related to COVID and influenza, and then do some typing and all that. So he figured out this pipeline, and he did it all. But she needed to add some, actually, phylogenetics. And I added this other step with making phylogenetics trees so you could see what was closer to what and so on. In this case, something very interesting happened that I worked with Allison, is her student, her PhD student. And actually, not even that, she was actually a technician, actually undergrad that became a technician. And she had some computer skills, and she took over. So I gave her the pipeline, it was already made, a little shiny app to look at the results, and I didn't hear from her for months then after that, so she did it all, she started to run all the results, and she did it, she took it off, and that was pretty good. That was actually what I was looking for, Gave her the pipeline and then the shiny app, I did my work, and then she took it off, and then she interacted with Emily, and then they did whatever they needed to do, and that was good for me, right? So that's another thing, you can have an existing pipeline and you can add on it. So that's what I mean with Nextflow. I use Nextflow for this. Nextflow, this person at the CDC built this pipeline, and I added it, with Nextflow, okay? So, you can also create an NF Core pipeline. In this case, there was this a tool called Copter. I don't know if have you heard it. It's for calculating microbial growth and in metagenomics. So this is established a kind of tool for doing that, and you can create an NF Core pipeline from it. So this is, in this case, this is the collaborator here, David Bernstein, and this is, again, her student. So again, I created all those modules, put them in NF Core. It took a little bit of work, did a little pipeline, and then she ran with it. And I haven't heard from her since. That was months ago, okay? So that was pretty good, and they run it. They do all sorts of stuff with it, and she has some skill in running things from the command line. You don't need to know. It's not that bad. She has some skill and some facility, and she can do it, okay? So that was great. Okay, so then this brings us to Sakera Platform, which is what Ken was talking about. There's people that know absolutely nothing, and they're afraid of the command line, or they don't want to do it or something, and there's also all these issues that can happen with the command line, right? And there's these things, and I interact a lot with the sequencing facility that does all the sequencing for genomes and nanopore and whatever, right? So then with the Sakera platform, then we can have a pipeline. We can put it on the platform, and then they can run it from the window, and they can go online and then run it. And that's what we did. So this is one pipeline that I have, and we have a second one now. And this is actually a very simple thing. It just concatenates fast queues. But it's obviously a very common request, right? And now the technician, she started doing it, and she does that routinely, concatenates, and does some basic QC. We added another one today that's just for transferring data. Something so simple, transferring data to the investigator. But now this person can go, fill up these things, and this is a person with some skill, can fill this up, and then the pipeline can work. Everything behind the scenes in this, in our HPC. And actually something interesting happened. Our HPC changed recently. They made some updates. And that would break everything normally. But then I fixed that behind the scenes, and they just kept running everything as usual, right? Because in a sense, this is independent of all the setup. Like this could be running on Amazon Cloud or on Slurm or whatever it is. So this is actually our group. So we have 3 bioinformatics people in our group, and then those two, so I don't know what I did, sorry. Here, excuse me. So this, the 2 people on the right, they're actually technicians in our group, and now they run those pipelines on the Sakera platform routinely. And again, I haven't heard from them in months. So yeah, so that works very well. So again, so you have the 2 extremes, the person, the bioinformatics person doing everything, to giving the power and the ability to the user, right, to run things. Obviously, to go back to the talk that this morning about reproducibility, this brings up some issues in that department, obviously, right, but one thing at a time. So So yeah, so this has worked very well, and I just wanted to mention, that the Secure platform and Nextflow had opened a lot of power, for making these pipelines and for providing them to the users and so on. So yeah, so last, I want to thank, and I just want to bring your attention to the collaborators that are there on the right. These are people that I've collaborated in the last year. And the ones in blue are people that I've used Nextflow and NF Core with. Obviously, there's many people that have benefited from these tools. So yeah, so any conversation you want to have on now or offline about that, I'll be happy to talk about your experience using this in collaborations. So thank you.
Thank you very much. That's a great talk. So you presented a couple success stories about students who really picked things up and took off of them, and that really appeals to me because I figure in bioinformatics course, the more computational savvy people we have in our community, the better everything is going to be. Do you have any sense, or have you identified any of the barriers that prevent people from running with projects like that?
Yeah, so I think it hasn't, maybe the thing is the person, in my experience, the person needs to know something. I ran into one situation where the student really was not comfortable at all with a command line, and that was actually it didn't really work, in that way, and actually one of her other students kind of took over in that project. So that's one thing, and that's maybe the Secure platform could work in that case for that situation. I also don't know, that's what I was telling you, Ken, earlier about teaching somebody to make their own pipeline. That happens with this copter situation. Maybe she's very ambitious, and I'm sure she wants to add something, but for her to do it and to add that, I just don't know what it would take. And me personally, I'm not sure. Would it need to take like a training, like several days training, a week-long course on Nextflow? Like it's kind of an open question for me. Yeah, thank you.
I kind of have a comment on that. I'm Max from HHMI. I think some of the comments that we heard from our collaborators in our institution when we're trying to implement them is like for Nextflow, you have to learn the DSL2, and all the syntax is for sure some learning curve. And sometimes they comment that NF Core is kind of have an opinion on the parameters you put in. So when you have samples that are not that standardized, it makes their makes it a little bit difficult for them to hack it through. And also, I wonder if you have experience like I think when running Nextflow, it usually creates a quite large disk folder that I don't know whether, like in your institution, you write other bash scripts trying to clear some of those intermediate files. Thank you.
Okay. So wait. So excuse me. So okay. So we have the intermediate files. We have the first one was what again? The first point?
There's still some learning curve.
Learning curve,
I think for
and
people.
the parameters. The second one was the parameters. What about?
Yes, I think some people think that the parameters are a little bit rigid, especially when you are doing something like not every other people are doing like with some special samples, for instance.
So some rigidity there.
Yeah.
Some kind of hard. Okay.
Yes.
Okay, yeah, so the learning curve, yes, there is a learning curve. I won't, deny it. If you want to kind of go and do like a pipeline and you want to do it, it's taken me a while to learn, and that said, the forum is very nice. There's a lot of support, and their documentation's pretty good, but there's no question that there is a learning curve, so it's learning a new programming language, right? So it's going to take, and I don't know what else can be done, right? Although there's a AI actually does, it is helpful, that tool, and I kind of used it a little bit, I started to use it more. But yeah, there's definitely like a learning curve. With the rigidity of the parameters, I don't know exactly. My experience, maybe we can talk more offline about that. Yeah, it hasn't been so much my experience about that, but there's definitely, I can see maybe that you might need to customize some things And if you have to get the pipeline and then make some tweaks, right? And I've had to do that, get an existing pipeline and then tweak it, because that's the only way to go get around that, right? Yeah, and yes, and what was the third thing? Work directories. There's ways to do it. Minus clean or something. Clean minus what something, right? Clean. Yeah. That would delete everything. But yeah, it makes pretty big intermediate things.
Yeah. One other thing that I'd mention about that, there is a Nextflow plugin called NF-Boost that will automatically clean up the intermediate files when something completes successfully. So that's one way to get around having a really large work directory. If you're working in a cloud environment, and this is what I did when I was building, we just put an automatic 7-day expiration policy on like the bucket that the data was going into, and so that way the data only sticks around for 7 days. On an HPC setup, you could replicate that with like bash scripts that auto-delete stuff in a certain path. And then on the learning curve, one other thing I'll mention, on the website training.nextflow.io, we have a free open source course for teaching Nextflow that's designed for anybody to pick up and teach people who are new how to use the basics of Nextflow.
Good to go. And it's very good. And you take it on your own pace, one coffee at a time, one lesson at a time. So I'm very aware that I'm the person between you and food and free drink, and I have been called, so I want to get out of here as well as soon as possible. So I'm going to try to make this just entertaining. So this is the first time I'm presenting this project. It started 1 year ago. This long title was given to me by ChatGPT because it thought that all the code and all the material we had there, this was the easiest way to put it in a slide. So I want to just, before getting into each of these words, I want to say that this is, what do you do after you use an NF Core pipeline? Because that is a big thing that is helping many people, but I'm working the Bioinformatics Core, and that is kind of the first step for us. Although now it's much easier, faster, we then need to use that data and answer the question with our collaborators about their specific project. So I want to just take 2 slides about why this is the title, long title, that ChatGPT chose to be the best. And the first is to explain how we work. So we work in 3 different ways. So we have consulting, and this is kind of the person in the group enter the room, or like we meet on Zoom nowadays and say, hey, this is my project, my data, my question, I need help. And it's like, okay, so you go one by one, choosing, customizing, trying to understand, okay, for this project, this tool, for this other tool. And we do this with many people. Then we have in the other side teaching. That is kind of the standardization of consulting, because you cannot just teach every single example to everybody. So we take what we learn from consulting on the community and say, okay, this is the materials we create on how to analyze RNA-seq data for this particular question. And all this material is out there. And then we have the middle part, that is where I focus more, that is actually, I think, that taking from both. Like, you need to standardize, but you need to be able to be flexible, and you need to be able to scale. And that is very difficult, I think, but this is why I love so much platforms in my day-to-day. So this is the first thing. And the second factor that I think that is very important, that kind of trigger this project, is that we are a group of 12 analysts right now, and so this is a plot that is showing how many projects we handled by year since 2015, because we are just gathering data since then. And so the y-axis is the number of projects. So it's 250 projects is a lot of projects with a lot of different questions from research. And it's not like we work with one specific institution. Like the colors are different institutions. And every institution actually kind of focus in different things. So now suddenly we have a decade or more of knowledge across 13 institutions and many technologies. Like we play with everything. So that is a lot of knowledge. And what do we do with that? So we are bioinformatics, this mix between engineers, artisans. I really like that slide. So I think that as bioinformaticians, we are trying to get into one side of our life, that is data, to the other side, that is insights, in different ways, reporting, apps, whatever. But in the middle, there is an architecture. And by the way, this slide is from Rob Syme, that as well is part of Sequera, last year's summit in Boston. It was not Boston, I think it was Barcelona. And in this architecture, there are different layers, right? So it's the layer of organizing your data, processing your data, and that is where I see Nextflow, and of course, Sequera, AWS, all these different tools. But there is this last step that is insight, like the learning, like, okay, the answer to the questions. And this is like the last part of like, how do we do this? Like for me, it's not clear nowadays. I think that people like are trying to figure out things. So I don't know, but just a question for you to think is like when you run your NF Core pipeline, once again for a different project, what do you do after that? You don't need to tell me, but what I have seen is that people will pull up RStudio, Python notebooks, and it will ask ChatGPT, it will go to a bioconductor, I don't know, tutorial or workflow example. In the past, I guess that we don't visit Stack Overflow anymore that much, but we like it because it's the input of ChatGPT, so we need it. Or probably you copy-paste from the previous projects. And that's fine. This is how we work internally many times. But I had this vision of like, OK, Nextflow solved the issue of processing data. And NF Core solved the issue of like creating the community and reusing that. So why not do we try to do the same after the analysis? And it's a much more complicated question because you need a lot of flexibility, but this is it. So VC Bio in the past was kind of like NF Core, I guess. But of course, we moved to NF Core Nextflow because it works much better, but we still have all this knowledge on how to analyze data, how to use this processed data from NF Core. What do you do with your expression data? What do you do when your project is this experimental design? So we have started last year to create templates, report templates, with guidelines, an example of codes for the technology we use the most. So RNA-seq is the most, because it's what still we have bigger and more complex RNA-seq, so things are getting a little bit out of control there. And then we have single cell, spatial single cell, ChIP-seq, and we are trying to build these reports where you can deploy this and have all this knowledge of our core, and of course from the community, because we work with the community, and you can jump into the report and start like saying, okay, I want to do differential expression, let's go and read chunk by chunk or step by step what I can do. And Just as an example, the last 3 slides is like RNA-seq, because it's the one that people still feels is generating a lot of data. So what you will get is like, okay, these are the dependencies that all these reports will need. They will have this quality assessment example of sequencing metrics, sample clustering, covariate analysis, differential expression, what batch correction. We have multiple examples. Group intervariability that, by the way, I haven't seen ever until I came back to the core. Like nobody, I think that it's the first time I have seen this type of analysis. Group comparisons, functional analysis, we know that. How to compare different analyses, there are templates for that. And how to do gene pattern analysis. And all this is using tools from many places, but commented with experience of people who are using it. So how you get that? There are multiple ways, but right now we are like having repositories for RNA-seq or for single cell that you can just download. We have a specific R package that is called BCBioR because it helps us to deploy this with one command, but you can just download that. And what you will get is examples documented for you, for the analyst, but as well for the people who is reading the report at the end. So you can see that like you will get some code at the beginning that you will need to point to the output of NF core pipeline, but there is a lot of notes in the code where, hey, we are using this, but just make sure that you're looking into that and these particular parameters because this is our experience. So there is a lot of code commenting on how the analyst can use this. But then as well is producing the final report where there is explanation on how you can interpret the results. And here, for instance, is one of the QC where like you should check your variability in your groups when you do RNA-seq because this seq2 is going to behave one way or another. So if you just go and run blindly an experiment, maybe it's okay with you, but if you have one group that has very little variability, another group that have a lot of variability, so you know your analysis is biased. So you need to take some action or just know that this is happening. And there is no way that a single analyst will know this, like every single detail, because that is, I think, what is beautiful from this particular project is that because there are many people running through different cases and then putting information back there, then you can just take over. So it's a way to transfer knowledge. And this is the group right now. There are like 3 main people in the platform building the core. We created an advisory team that because I really want this to merge pretty good with NF Core. We invited Phil and Geraldine, who are hardcore members of Nextflow and NF Core, and Lucas, who is a good, as well a strong member of single cell and R community. And we hope that we can put this out there and then start getting feedback from people. So we create kind of this massive knowledge, structured knowledge base for some of the analysis. And that's all. I just mentioned like communities and institutions that are supporting this work, and that's all. Thank you. We did it.
Thanks a lot, Lorena. I was wondering if you could go into the decision-making process. Like the NF Core differential abundance workflow has some overlap with the RNA-seq workflow that you described. Can you talk a little bit about how you decided to redo that? And yeah.
I like how I think that both of them are useful and can coexist because this is targeted to cases where, like you don't have like a platform where data is coming the same way and you really know exactly how to analyze it. And anyway, this platform is like differential abundance is good for a first round, but this is targeted more like for the day-to-day researchers that we find out there, and as well to teach, because one component that for us is important is analysts knowing the very detailed edge cases of each of the analyses. So it's something that you won't have a differential abundance analysis because I think that you will use that more in a platform pipeline situation or for a first maybe interaction with the data. And this is more for like the analyst looking at the data step by step because want to make sure that it's covering all the different angles. And ideally, this is going to grow beyond differential abundances.
And a question for everybody. We're talking about cleaning up the work directories and using a bash script to do it. Anyone doing bioinformatics has got a collection of bash scripts in their pocket. When do you take the time to invest to convert those bash scripts to Nextflow code so that they can be run that way, and why would you go about doing that?
Is this a prepared question? Okay, so because we had this discussion, and I went like, you know what, next time I'm in that moment of my life of deciding to do this, the bash script or the pipeline, I'm going to try to do the pipeline. So I had this like subsampling example of trying to, that everybody does, that subsampling FASTQ files. And I went to Sequera, used AI, told exactly what I needed, and it got me 80% there. The 20% was like 15 minutes, and in 30 minutes, I had something working in Sequera. So I would say nowadays, I don't do bash anymore. I just invest my 30 minutes on that because then the rest of the group can use it. But maybe that is an experiment end of equal one, so maybe it fails next time.
Hey, Lorena, so I just have a question. How do you deal with versions of packages in the templates?
Yeah, and so that part is the one that we are not handling that much. So we have like a soft versioning of basically if you're using Python or Bioconductor. But we are, ideally, I think that my next step will be to containerize, at least give the opportunity to use containers for that. But again, we are very much focused on the decision making of the scientists than having all the packaging, all the package and tooling installed so you can go through all the templates. More like, oh, I'm very interested in this analysis. And I just want to know right away what are the tools and how to use the parameters for that. But it's something that we need to get there as a community.
At our place, we deal with it with Docker images of our studio server and run them with Singularity on the cluster. So that's how we handle that. Yeah, I think we have maybe 5 minutes left or so. If anybody has any more questions, come to the mic, or if you want to go up to the table and discuss, whatever works for you. But thank you for coming, and thank you to the speakers for a great session.
Okay. Mission accomplished.
Yeah.
Is she
Yeah, she was around. She came before, but then she left. But yeah, she's around.
I got a quick question for you. Like,
Thanks. So you guys are building out, like I'll use your bulk RDC. You're building out some steps beyond the basic differential expression. Down the road, would you ever consider pushing those out?
Yeah, there's an option in that for
It is so we are already talking about all that.
no. So
We are
with the
getting
Nextflow
stuck
plugin,
into the.
you can
We are not
enable
sure
the
if
plugin
it
in any
can be a
Nextflow.
pipeline. Because the idea is
What the plugin
this
system does.
this
It lets you
specific.
kind of add things to
Actually,
your
started in the studio. Because it's what
Yes, exactly. So one of the features in that plugin
we cannot
is
use that.
auto-delete.
Say that, I think that probably there is going to be some cases.
Yes, just for that.
Pipeline,
Not everything. Yeah, it doesn't make. It's not cost effective to make everything completely automated all the time. So you got to pick your targets. That makes sense. Thanks. That's great.
I'm on the Bioinformatics Core Facilities, and I'm actually pretty heavily adopted in Core. I'm actually full development.
Yeah, that's going to be different.
But I do have a question. How have you found ways to find additional support?
Yeah, so usually it comes down just to reading the documentation carefully. A lot of Nextflow pipelines want you to submit a sample sheet, and that sample sheet will have like the list of these are all that my data is going to use, and that's kind of the way this data source works.
Yeah, so there are two things happening. One is that we have some funding coming from grants. That is
That sounds like a question you
really
might
do
have to
analysis for like
forum.
other things,
And
but
then
we
I
use
can help you
some
out.
part
And
to work on that because at the end it just
forum
makes
is
things easier. So taking that, and then
Okay, we
we
have
charge
asked for help.
like by project hours. So we kind of try to make sure there is always a small percentage for each project dedicated to that. Like we always get RNA6, so there is always something inside that project that can align with developing that part. So it's a lot. To be honest, this is like what I spend a lot of my time, like trying to have this dashboard of tasks that very well fragmented, that can be like put it in different projects and coordinating all that. Because the majority happens like that. Like very small happen because the grant, so there is more time, but the